Hi guys, welcome back. So today's lesson is going to be on the circulatory system, and this is going to cover kind of the one you think of in humans as well as alternate circulatory systems in other organisms. So let's get started. So first, just with some basics. So essentially, in an organism, exchange has to happen in every cell. We need to remove waste products like CO2 and import required resources like oxygen and nutrients. So this has to happen for every cell in an organism. And, for small molecules, diffusion is kind of the preferred means of transportation, in and out of the cell. But the thing is, diffusion is slow, as the time it takes to diffuse is proportional to the distance squared. So it grows really fast compared to the distance. So, since exchange has to happen in every cell, it's pretty easy for unicellular organisms to just do all their exchange through diffusion. They've only got one cell, so there's really not a lot of distance to transport between. But direct exchange with all of the cells is impossible for multicellular organisms, which is why we need circulatory systems. So there's kind of two routes we can go about this. So the first is a simple body plan that maximizes contact with the outer surface. And this is found in cnidarians like jellyfish and flatworms. The alternate kind of route is a circulatory system with fluid reaching near every cell. And this is often linked to gas exchange, and this is what you see in most animals. So, kind of the simplest body plan we have is a gastrovascular cavity. Essentially, what it is it's kind of like a big sac with one opening. And this sac might be like folded into different shapes. Like you see a jellyfish and you wouldn't think that like there's a sac within kind of its body. But, essentially that's what it is. When you go through the mouth, there's a bunch of canals, but those are all one continuous space. And the purpose of a gastrovascular cavity is to distribute substances throughout the body and also digest. So, we've talked about the hydra already, but hydras, they live in ponds and they've got this pretty simplistic gastrovascular cavity where they can have their prey in it and digest it and then just eject whatever they don't digest out of the mouth. And it's this gastrovascular cavity that, kind of conducts exchange with the environment. And, same thing with these flatworms, right? So, this flatworm, all this orange stuff is the gastrovascular cavity. As you can see, there's like a lot of folding and whatnot, but it's still one continuous space. All right, so now let's talk about circulatory systems. So, circulatory systems are going to have three main features, which is a circulatory fluid, interconnecting vessels, and a muscular pump or heart to increase hydrostatic pressure, which is the pressure essentially of, like a fluid pushing out. So, in an open circulatory system, the circulatory fluid is hemolymph, which is also the interstitial fluid. So, in humans, we have blood, right? But we also have interstitial fluid, which is the fluid between our cells, and those are two discrete things. But, in an open circulatory system, hemolymph kind of serves both of those functions. So we see this in arthropods and some mollusks, and its version of interconnecting vessels is a system of sinuses, which are spaces around organs. And crustaceans are going to have a more complicated open circulatory system. And kind of the benefits of an open circulatory system is it has lower hydrostatic pressure, which means it takes less energy to maintain this type of circulatory system. Like, you don't have to spend so much energy pumping the hemolymph to really, really large pressures. Plus, it can have other functions, like spiders. They use that pressure of the hemolymph to extend their legs. So, a closed circulatory system is kind of what we know as humans. It's what we have. It's what, like other mammals and whatnot have. So, instead of hemolymph, we have blood. And this is not the interstitial fluid. Those are separate things in a closed circulatory system. It's found in annelids, cephalopods, and vertebrates. And we have blood vessels that go into tissues and organs. And the benefits are that high blood pressure is good for big and active animals. So, we move a lot and we're big. So, that high blood pressure kind of makes sure that the blood gets everywhere it needs to be. And you can kind of see this in that some mollusks have an open circulatory system, but the most active ones have a closed circulatory system to support all of that movement. And in general, in a closed circulatory system, blood distribution is better regulated than in an open one. All right, so now let's just talk about the basics of the cardiovascular system in vertebrates. So I've just made this kind of basic diagram to show how things go. So you have your heart that's going to pump blood to your arteries and then arterioles, and that's going to be in an organ. And then it's going to go through a capillary bed and return through the venules and the veins. So, basically, you can kind of see that the arteries and the veins are the thickest arrows, and that means, I did that so that symbolizes that they're the biggest, blood vessels by diameter, and arteries and arterioles are going to have thicker walls than venules and veins because, they have to carry more pressure, like the blood, it just got pumped out of the heart, so there's a lot of pressure in it still, and you have to have a lot of like muscle and elasticity within the walls to, kind of keep that structural integrity. Capillaries are going to have very thin walls, they're going to be very thin themselves, and then, our blood returns. So, kind of just some notes. Arteries and veins differ by the direction they carry blood. So, in humans, with the exception of like the pulmonary artery and the pulmonary vein, arteries and arterioles are going to carry more oxygenated blood than venules and veins. And just something else, portal veins are veins that carry blood from a capillary bed to another capillary bed. So, for example, the hepatic portal vein carries blood from your digestive system to your liver. So, yeah. And then, your heart has various chambers. So, at the top, we have atria, which receive blood, and ventricles are at the bottom, and they pump blood. So we're going to get into specifics about all of these. All right, so there's a couple types of circulation within circulatory systems. So first we're going to talk about single circulation. So you find this in sharks, rays, and bony fishes. And essentially it's called single circulation because the blood really just goes through everything once and then returns back to the heart. So first, the heart pumps, like this oxygen-poor blood through the artery, and that makes it to the gill capillaries where it's oxygenated, and then that oxygenated blood passes through the body capillaries where the oxygen gets used up and then returns through the veins to the heart. So there's going to be relatively sluggish flow in single circulation because one pump of the heart has to get all that blood this full circle, and a lot of the animals that use single circulation use their muscle movements to help blood move around. So all these fish, they're swimming constantly, and that muscle movement can kind of like squeeze that blood through all of these blood vessels. So the alternative is double circulation. There's two sort of variants here. So we find this in amphibians, reptiles, mammals, and we have two circuits that make up our double circulation. There's a pulmonary circuit or a pulmocutaneous circuit. So, essentially some animals, they also use their skin to breathe, so to say. And in that case, they would have a pulmocutaneous circuit. But, we humans, we have just a pulmonary circuit. And we also have a systemic circuit. as our other circuit in double circulation. And essentially, the pulmonary circuit or the pulmocutaneous circuit has a right side that pumps oxygen poor blood to gas exchange tissues. Um, and just as a note, some amphibians and reptiles are intermittent breathers. That basically means they don't have to be breathing all the time. And this has to do largely with kind of the specifics of how their circulatory system is set up, which we'll talk about very soon. And then the systemic circuit is just basically it has the left side of the heart pumping oxygen rich blood to the body. And since we get, have these two separate circuits, we have more vigorous circulation than single circulation, and thus higher blood pressure. All right, so now let's talk about those adaptations for intermittent breathers. So, as you saw here, there were a few differences between, like mammals circulation and amphibian circulation, and you can see that like right here, the heart of an amphibian actually is not like completely divided the way a mammal heart is. So this is actually pretty advantageous for them. So for amphibians, they use the pulmocutaneous circuit, and they have a ventricular ridge instead of a septum like mammals do, like a complete septum. And as a result of this ventricular ridge, about 90% of blood goes where it should, so to say, go. So there is like some mixing of oxygen rich and oxygen poor blood, but this is advantageous for them because when they are underwater, they can stop most of their blood flow to their lungs. So this is something nice for them. And, in turtles, snakes, and lizards, there's also some adaptations. They have an incomplete septum, so it's more than a ventricular ridge, but it still doesn't divide the ventricle, doesn't divide the heart completely. So, there are two aortas that lead to systemic circulation. So we got one here, and basically this allows for control over how much blood goes to the lungs and body. So when these animals are like underwater, they can't use their lungs to breathe, so it wouldn't make sense to divert blood to the lungs. So they would just divert it away until, they would need to use their lungs again. And for alligators, caimans, and crocodilians, it's kind of the same story. They, however, have a complete septum, and they have a place where the pulmonary and systemic circuits actually connect, where the arteries exit the heart, and their blood flow can be diverted from their lungs when they're underwater. So you can see that like these adaptations make it so they don't have to be breathing all the time, which is really helpful because these animals spend part of their time in water and part of their time on land. And just as a note, birds and mammals are endotherms, and they need 10 times the energy as some other animals, which means they need powerful hearts. So yeah, birds and mammals have very similar kind of like heart structures as opposed to like these animals. All right, so now let's talk about the mammalian circulatory system. So we're just going to go through this picture really quickly. So we're going to start at number 1, which is essentially we've got this oxygen poor, thus it's blue, blood going in the ventricle from the atria, the right atria going into the right ventricle, and then the ventricle is going to pump the blood into the pulmonary artery, into the lungs, and then that's going to come back through the pulmonary vein and into the left atrium, and this is the most oxygenated this blood is going to be. And it's going to go to the left ventricle, get pumped out into the aorta, which delivers it to the rest of the body, and it comes back through the superior and inferior vena cava into the right atrium, and it starts to cycle again. So just a little bit more about the heart. So I kind of talked about how it moves through everything via pumping. So we have what we call a cardiac cycle, which is this cycle of like pumping and relaxation that makes sure blood goes where it needs to go. So essentially, diastole is relaxing of the muscles. Systole is contraction of the muscles, and we start with both of them being relaxed, and the atria and the ventricles getting filled up, and then we have atrial systole and ventricular diastole, basically the atria emptying all its contents into the ventricles, and then we have ventricular diastole and ventricular systole and atrial diastole, and that blood gets pumped out. So we have this cycle going over and over again to deliver blood, and in the process we need valves between the different chambers of the heart to make sure that blood's not going the wrong way. So we have, as you can see, we have our tricuspid valve here, and this valve is in between the right atrium and the right ventricle, kind of in the same place on the other side. We have the mitral valve, and it's also called the bicuspid valve sometimes because it has two cusps, whereas the tricuspid valve has three cusps. And then we also have a pulmonary valve here and an aortic valve here. And just kind of like general names for these valves: atrial ventricular valves, these two, this one and this one, these are called atrial ventricular valves because they're in between the atria and the ventricles. And then these are called semilunar valves, I believe, for their shape. And just some kind of details on the anatomy. These valves are held back by these things called chordae tendineae. They're these kind of like fibers, and they're held to these muscles called papillary muscles, and this just prevents the valves from turning like inside out due to the pressure of pumping. So, yep. And then just something else, cardiac output, which is a calculated number, is the heart rate times the stroke volume, which is the amount of blood pumped per beat. And this basically just shows like per minute, depending on what you're measuring your heart rate in, how much blood you're pumping. And this is about 5 liters. And when you listen to the heart with a stethoscope, you're going to hear a lub-dub sound, and the lub is the recoil against the atrioventricular valves. So basically, the atrioventricular valves are closed, and then the blood is like thudding against it, while we have contraction, and then the dub is the closing sound of the semilunar valves here. And you might hear a heart murmur, which is where blood goes backwards through a valve, which is not good. Alright, so here's just more specifically on the cardiac cycle. So we have our two phases of systole and diastole. So during systole, we have isovolumetric ventricular contraction, which is essentially where we have no change in volume because all of our valves are closed, but the ventricles begin to contract. And then we have ventricular ejection, where our valves here to the pulmonary artery and to the aorta are open, and thus, the ventricle is able to contract and actually like get smaller and squeeze the blood into those vessels. And that completes systole. And then we have diastole. There's isovolumetric ventricular relaxation, and this is basically when all the valves are closed but our ventricles relax. And then we have ventricular filling, this kind of broad phase where we start filling the ventricles, and basically our atrioventricular valves are going to be open while the blood kind of filters through the atria, goes in, and then we have the atria actually contract to squeeze more blood in. And then that is basically a cardiac cycle. So now you may be wondering how our body kind of coordinates this like very orderly system of contractions and whatnot, and it does this by using electricity. So here is just kind of like the conducting system of your body. So we have a sinoatrial node as our starting point, and this is often called the pacemaker of the heart. It's basically the like frequency of signals sent out by the SA node determine how fast your heart is beating. And then you have the atrioventricular node right here in between kind of the right atrium and the right ventricle. And then that leads to the bundle of His in the middle right here, splitting off into the right and left bundle branches and ultimately becoming Purkinje fibers. So these cells are going to conduct this signal through gap junctions. It's going to be really, really fast. That's why your heart is able to synchronize its beating so well. And it's really fast, kind of with the exception of the atrioventricular node, which purposefully delays contraction, like relaying the signal. So the ventricles contract after the atria. And yeah, basically we can just see what happens in this picture right here. The SA node is going to kind of discharge a signal. AV node is going to get said signal, and it's going to spread through the entire atrium at this stage, and then it's going to continue on in the order I just talked about. So yeah, basically that's the conducting system of your heart. And you like contraction is pretty much simultaneous in the right and left halves of the ventricle, except for the bottom of your heart will actually contract a little bit sooner than kind of like up here. So there's like a toothpaste like effect with that. Like when you squeeze toothpaste, you're squeezing from the bottom of the tube, and like that part gets squeezed first, but the parts up top get squeezed soon after. Okay, so now let's talk about EKGs or ACGs and how to read them. So you've probably seen this type of picture before, but each of these waves actually means something. So the way an ECG or EKG works is there are these leads placed on you, and they detect like shifts in the electrical activity in like the fluid of your body. So, obviously, because your heart uses electricity, there's going to be kind of like these little effects afterwards, and you can detect those through your body fluids, and that is the point of an EKG. So, we have our P wave. It's supposed to last less than 0.11 seconds. And oh, by the way, one of these grids of 5 represents 0.2 seconds. And this P wave is atrial depolarization. The QRS complex as a whole is ventricular depolarization, and it does coincide with atrial repolarization, so that's why we don't really see like a bump for atrial repolarization. But anyways, just specifically, the Q wave is when the septum, in between the ventricles is activated. The R wave is when the ventricular free walls is activated, and the S wave is just where everything else is activated. And then we have the T wave, which is bigger than the P wave, and this is ventricular repolarization. And then we have just something to consider is the ST segment, and or from here to here should be straight, and if it's elevated by more than 1 millimeter, that could be a sign of early ventricular repolarization. So if you've actually heard, like if you've been watching Grey's Anatomy or something, you've heard them say something like ST elevation or like STEMI in relation to a heart attack. They're talking about, a heart attack where there is an elevation of this segment. And then the PR interval, between like here and here is the conduction time of the AV node, usually between 0.12 and 0.2 seconds. And the QT interval right here is the time of total ventricular depolarization and repolarization. So yeah, it's not crucial for you to remember every single little thing about this, but just know that, everything kind of corresponds to an activity going on within the heart. All right, so now let's talk about blood vessels. So we're just going to look at the wall compositions of different blood vessels. So we have our arteries, our veins, and our capillaries. So you see that for the arteries and the veins, we have like the same layers, but they have different thicknesses and slightly different makeups. So first thing to note is that the lumen of the artery is much skinnier, because the walls of an artery are 3 times thicker than those of veins. And also note that the veins have valves because, like when your heart is pumping blood into the arteries, just that pressure of the blood is enough to keep it going the right way. But once it gets to the veins, it's pretty slow. So there needs to be valves to make sure that the blood isn't going the wrong way. And arteries are going to be very elastic and very strong, so they have a very distinct tunica media layer as opposed to the veins, right? You see how thick it is, and it has lots of elastin to kind of keep those walls elastic, so they can kind of like respond to the pressure of the blood being pumped through them. So yeah. And for capillaries, there's a basement membrane around a kind of like sheath, like as a sheath around endothelial cells. So yeah, that's kind of the basics of wall compositions. So now let's talk about velocity and pressure. So obviously I mentioned that blood gets pumped through the arteries really quickly, and by the time it makes it to the veins, it's pretty slow. So in physics, there is this equation, the equation of continuity, A1V1 equals A2V2. So this is essentially describing the flow of a fluid through some kind of like pipe or tube. So obviously in our context, our pipe or tube is a blood vessel, but it's saying that the cross-sectional area, so if you were to just like kind of like cut a blood vessel, it's like this area right here, that the blood can flow through, times the velocity or like the speed of the blood through that point equals the cross-sectional area of another point times the velocity of the blood at that other point. So what that's saying is basically the larger the surface area of like the vessel, the slower the blood is going to flow. So we can kind of apply this analysis to these graphs. So you might think of capillaries as having the smallest surface area, or like smallest cross-sectional area, and you would be right to some degree. So basically, capillaries, they have a small cross-sectional area when we're just looking at like one capillary compared to the cross-section of one artery or one vein. However, there's so many capillaries within your body that their combined cross-sectional area is actually pretty huge. So you can see in this first graph here, the cross-sectional area of like arteries and veins is like nothing compared to the cross-sectional area of capillaries. And according to our equation of continuity, that means that blood has to go pretty darn slow through those capillaries. So you see, blood's going really fast through these arteries, and it just really dips around the capillaries, and it kind of goes back up through the veins. And obviously the pressure is fading over time, which is kind of causing this like lower velocity in the veins. But here's just kind of what pressure would look like in these blood vessels, and just in general, the further we get from like the pumping of the heart, the less pressure there is. So yeah. And basically just some things. So we have a systolic pressure and a diastolic pressure. Systolic pressure is just the pressure you can feel when your heart is undergoing systole, and then diastolic pressure is your blood pressure when your heart is going under diastole. And basically your systolic pressure is just caused by like the beating of your heart, and the diastolic pressure is caused by the artery walls snapping back after they've been kind of like expanded by your pulse. And and then like if you've ever gotten your blood pressure from the doctors or something, they give you like a number over a number, and that's your systolic pressure over your diastolic pressure. And some things that could kind of change velocity and pressure: vasoconstriction, vasodilation are when your arterioles narrow and dilate respectively. Endothelin is kind of like the mem for vasoconstriction. It's a protein that causes your arterioles to narrow, and nitric oxide is a gas that causes your arterioles to dilate. And something else to note is that gravity affects blood pressure. So, like our heads are above our heart, and that means like the blood pressure is going to be significantly lower in our brains as it is like straight out of our heart. And think about something like a giraffe. So giraffes have long necks, right? So their heart is really, really powerful in order to get that blood up to their brains. And then this picture here just shows how you can measure your own blood pressure with a sphygmanometer and a stethoscope. Basically, just tighten the cuff so your blood flow is cut off, and then you place your stethoscope in that artery, and you loosen the pressure until you hear a sound, and then you note that number. That's going to be the systolic pressure, and then you keep on loosening until you hear no sounds, and then that number is going to be the diastolic pressure. And blood pressure is typically 120 over 80 or under. All right, so now let's talk about capillaries. So something very interesting is that while you have so many capillaries, only 5 to 10% of them are in use at a time. And this is regulated by vasoconstriction, vasodilation, and these things called precapillary sphincters. So you can see them in this picture here. They're these sphincters kind of like before a capillary bed. And depending on whether or not they're constricted or relaxed, blood can or can't go through those passageways. And then we also have these things called thoroughfare channels that are always open. So yeah, and just some examples of when we might want to alter the blood flow to a location. So if you get injured, you can notice that your injury is going to get inflamed. It's going to get kind of like red and swollen, and that's a sign of more blood flow to the area. So, if we get injured, our immune system is going to have cells that release these things called histamines that are signaling molecules, and that causes vasodilation at the site, so that your body kind of is able to recruit a bunch of, like white blood cells and whatnot to the site to start healing and defend against pathogens. So yeah. And then there's also increased blood flow to your intestines after a meal, because it's not really realistic to be spending a lot of your blood supply on maintaining your digestion when there's not anything to digest. But after you eat, there is stuff to digest, so you're going to get a little more blood flow to your intestines and your digestive system. So here are like 3 types of capillaries. You're going to see there's continuous, fenestrated, and sinusoidal. So continuous capillaries, they're going to be like decently non-leaky. And then, oh, by the way, capillaries are going to have one kind of like complete-ish layer of endothelial cells. And they're also going to have these cells called parasites kind of periodically around them. So, continuous capillaries are continuous, and there's not too many gaps, but there are pores. And fenestrated capillaries, they have these fenestrations, which are these like gaps, and that just makes them more leaky than our continuous capillary. So you might see this in places like the kidney. And sinusoidal capillaries, they have a lot of like holes, right? Like they have a lot of gaps, and this is kind of found in areas of the body where you're like filtering the blood, or you need to access it to liver directly, like the spleen or the liver. So yeah. All right. So now let's talk about capillaries and lymph. So in your capillaries, you have what we call Starling forces, and then there's a hydrostatic force or pressure, which is like the pushing out pressure, and then oncotic is like kind of like the pulling in, or like osmotic sometimes it's called. So essentially, like our hydrostatic pressure our within our capillaries is going to be like our blood pressure. Our oncotic or osmotic is mainly going to be the pressure of like proteins like albumin within our capillaries that's going to want to pull water in. So yeah, it's maintained blood plasma proteins like albumin. And essentially, in our body's overall blood pressure is going to be greater than osmotic pressure, and we're going to lose some fluid into this interstitial space, which is recovered by the lymphatic system. So, yeah, you can kind of see the net movement or like the net pressure at the arterial end. It's going to be plus 10 millimeters of mercury. And then the net pressure inwards at the end in the venous end is about negative 7 millimeters of mercury. So, we're going to have net movement out. That's what our lymphatic system is for, to basically, like recover all that fluid we're losing. So now let's talk about blood. So blood, kind of the common saying is that it's formed elements like cells and cell fragments in plasma. And plasma is kind of complex. It has a lot of things, right? So if you've ever like gone donating blood or something, you might have been donating plasma and not blood. So basically, plasma is when you remove like the cells from bloods. So you end up with this kind of like yellowish liquid. So you can see right here. It makes up about 55% of your blood, so it contains proteins, nutrients, metabolic waste, and electrolytes. Also, a bunch of plasma proteins, so there's albumins, immunoglobulins, apolipoproteins, which carry lipids in your blood since they're not soluble. Fibrinogen, which is like a precursor to blood clotting. Hormone transporting plasma proteins, and then transferrin. Right, so those are just like some examples of plasma proteins. Here's like a picture with a few more. And then there's serum, which is just plasma without clotting proteins. Don't get these two mixed up. And then we have cells. There's erythrocytes or red blood cells. There's platelets, which help like with blood clotting. And then there's leukocytes or white blood cells. So yeah. All right, now let's talk about blood clotting. So, there's kind of like three big steps. So you have a enzymatic cascade that's going to ultimately convert prothrombin into thrombin. And then this enzyme thrombin is going to convert fibrinogen, which is like the inactive soluble form of fibrin, to fibrin, which is not soluble. And fibrin is going to make a clot that's going to ultimately kind of like plug the wound up. And then like this is kind of like the long-term solution, but immediately after you get a cut or something, like you're going to have these platelets floating around in your blood, and they're going to like plug up the wound to basically like make sure you're not losing blood too much. And then yeah, so fibrin is kind of going to like seal the deal, but it takes longer to get it ready. And just kind of like a piece of, I guess, like trivia or information, is that vitamin K is really important in blood clotting. All right, so now let's talk about diseases. It's just like a slide with some diseases that you should probably know about the cardiovascular system. So there's atherosclerosis, which is where arteries harden because of plaque, which are just fatty deposits. So you can see in this picture right here, you've got this plaque right here, and then the lumen would have been much larger. It would have been this like entire space if it weren't for the plaque and the thrombus that are blocking the way. So essentially, this has a lot to do with cholesterol. So there's two types of cholesterol that we need to know here. There's LDL, low density lipoprotein, and HDL, these are cholesterol carrying proteins, my bad. HDL, which is high density lipoprotein, And LDL delivers cholesterol, HDL picks up cholesterol and returns it to the liver. So LDL is like the quote unquote bad cholesterol because it like drops off the cholesterol, and HDL is like the good cholesterol because it kind of picks it up from random places in your blood. So you want a like low LDL to HDL ratio, and then there's like certain diets like the Mediterranean diet that can kind of facilitate that, good for your heart. And basically, with atherosclerosis, a thrombus can form. And a thrombus, is just kind of like this chunk that can move in your blood and block different, blood vessels. So it's very bad. And then angina pectoris, which is just chest pain, is like a symptom of atherosclerosis because it's kind of basically like parts of your heart or whatever aren't getting enough blood. So yeah. And then a heart attack or myocardial infarction is where coronary arteries are blocked. Coronary arteries are the arteries that supply blood to your heart. And if they're blocked, heart muscle is going to die due to a lack of oxygen. And that's what a heart attack is. And then, a stroke is when nervous tissue dies due to a lack of oxygen because of a clot, which would be like an ischemic stroke, or because like there's a like bleed in the brain, and that would be a hemorrhagic stroke. So either way, lack of oxygen. And kind of like a fix for atherosclerosis, would be inserting a stent. So basically, you have this like stent, usually made out of like platinum or something, and it's going to be inserted. It's going to be around the balloon. It's going to get inserted, and it's going to be kind of propped up by inflating the balloon. The balloon is removed, and then now you have the stent holding open an artery. And then, technology for this is actually really good these days. So they could literally do this entire surgery like through a cut on your thigh. So, like definitely minimally invasive surgery techniques are good for this. And that is it. I really hope this was helpful. I know that was really long, but questions about the cardiovascular system usually are not so difficult on both, USABO Opens, Science Bowl, USABO semifinals, and whatnot. Think there's just like a lot of information, but once you master it, I think it's definitely going to come in handy for you. So yeah, stay tuned for more anatomy and more question practice. And if this helped you, please leave a comment or a like and subscribe for more content. Thank you.